So the Centers for Disease Control and Prevention, the famous CDC, has categorized the emergent resistant Neisseria gonorrhea as an urgent public health. And this is like the highest category of threat in terms of antimicrobial resistance. So today we will discuss this emerging problem of an important problem in gonorrhea and implications for public health with really outstanding experts in the field. So the objectives of the podcast is to review the clinical implications of resistance to B-lactams and quinolones in Neisseria gonorrhea, discuss the mechanisms of resistance in these organisms, and provide some insight into the public health implications and novel therapeutic approaches to treat these resistant gonococcal infections. Welcome to Editors in Conversation. This episode is brought to you by the Antimicrobial Agents and Chemotherapy journal available at asm.org/AAC. I'm your host, Cesar Arias, Editor-in-Chief of AAC, and this podcast is supported by the American Society for Microbiology, which also publishes AAC, among other outstanding journals. If you're planning to publish in AAC, which is one of the top journals in the field, and you are an ASM member, you'll get actually a discount of up to 50% in publishing fees. Visit asm.org/membership to sign up. And don't forget to check our latest issue of AAC available at asm.org/AAC with outstanding papers on mechanism of resistance, pharmacology of antimicrobial agents, epidemiology, and clinical therapeutics, among others. I want to say today that we actually have a new section available in the website, which we're putting a collection of our published new perspective of antimicrobial agents when we are reviewing current thinking about antimicrobial agents and novel agents that are about to be approved by the FDA. These have been really great resources, and now you can find them there as a full collection. We include antibacterials, antivirals, antifungals, and that is a great resource for information. Georgie and I, I'm very, very honored to have these two distinguished speakers today, guests of the program. Dr. Bill Shafer, who is professor at the Department of Microbiology and Immunology at Emory University School of Medicine. He's the co-director of the Emory Antibiotic Resistance Center and is being named Dean's Eminent Investigator at Emory University School of Medicine. And Yonatan Grad, who is the Melvin and Geraldine Glimcher Associate Professor of Immunology and Infectious Diseases at the Harvard T.H. Chan School of Public Health. Gentlemen, welcome to the program.
Thank you very much. Glad to be here.
Thank you, Cesar. Pleasure to be here.
So great to have you guys experts in different aspects of gonococcal infections. Let's start, Bill. Let's start with you.
Sure.
Let's talk about the gonococcus. Why is where is it coming from? Something about the biology of the gonococcus.
Okay, so to me, Neisseria gonorrhea is an absolutely fascinating pathogen. It's a strict human pathogen that causes the sexually transmitted infection, and we do call it STIs now, term gonorrhea. And gonorrhea can come in many different forms, the typical genital tract infection in males and females, but it can also cause rectal infections, oral infections. It can cause very severe upper tract infections in women. And it can also get into the bloodstream and cause a disseminated gonococcal infection. And what's interesting is, and we call that DGI, DGI seems to be making a reappearance now. We've had some outbreaks in various parts of the country. Many infections are asymptomatic, especially in women. Now, to me, there's several interesting biological features of this pathogen. It has the capacity to resist a number of innate host defenses that are brought to bear. People can get multiple infections because there's no immunity to reinfection. We desperately need a vaccine, and now that we're learning more about immunology, human immunology at the mucosal site, we're hopeful for that. But what is also very interesting to me is that this pathogen is a chameleon. it has about 100 phase variable genes, so it can redecorate its surface by changing lipooligosaccharide structure, pilus antigenic type at very high frequency. So for today's conversation, I guess we're going to be focusing on antibiotic resistance. And beginning in the late 1930s, when sulfonamides were introduced, we should have learned a lesson, because within 3 to 4 years, the level of resistance to sulfonamides increased dramatically. Fortunately, penicillin was coming in, and it could be used, and it was actually used very well for, I guess, over 40 years. But every time a new antibiotic is brought into clinical practice, sooner or later, we're going to have isolates that are clinically resistant. And what's worrisome now is that the overall pipeline for antibiotic development has certainly decreased. So we're running out of options. There's a couple of antibiotics we can talk about later that are in phase 3 trials, but we do have a problem that right now in the US and other well-developed countries, we are down to a single antibiotic for empiric therapy, and that's ceftriaxone, which is a third-generation cephalosporin. And we have had strains that are clinically resistant to that. In
So,
fact,
Bill,
even in the US.
we will talk a little bit more about this. So I just want to get Dr. Grad here, who is also an infectious disease physician, and tell us. I remember my one of the last patients that I saw with a very severe gonococcal infection. The patient actually had endocarditis. So people say, well, why the CDC has this as an urgent threat if really gonococcus does not, or they don't produce fatal infection. So what would you say to that comment, and it could expand about the clinical manifestation of gonococcal infections?
Sure. So I think Bill did a fantastic job introducing gonococcus and the infection caused by Neisseria gonorrhoeae. And to expand on that a little bit, give you a sense of the scale of the problem. So in the last year for which we have reported data, 2020, there were nearly 700,000 infections reported in the US, with a true incidence thought to be at least double that. And that's in part due to what Bill was describing, that infections at a number of different sites, the cervix, the rectum, and the pharynx, tend to be asymptomatic. Infection at the urethra tends to be symptomatic. But it's an extremely high burden of disease. In fact, it's one of the most common bacterial infections, the second most common reported STI, with chlamydia being number one. But a huge volume, and it's going up. We had a historic low in incidence in 2009, and we're now more than double that number, and it continues to increase, both in the US and globally. We've seen similar increases in countries where we've seen data, the UK and Australia. What happens if you have untreated gonococcal infection, right? So going back to the pre-antibiotic era, or what we worry about and why it's such an urgent threat. Well, for men with urethritis, you end up with scarring and strictures in the urethra. And if you ever have gone into one of these museums of medical devices and seen the urethral dilators that people had to use for men with this consequence of gonococcal infection, you can understand why people might think it's urgent. That is in one situation. The other is in women, the severity of an infection that Bill mentioned with pelvic inflammatory disease as it ascends through the female genital tract was associated with scarring of the fallopian tubes, ectopic pregnancy, and infertility, and all sorts of consequences from that. And also, as Bill was mentioning, you can get disseminated gonococcal infection where gonorrhea goes into the bloodstream, and you can get the kind of endocarditis case that you were describing, Cesar. So, there are certainly severe consequences of infection that we've seen. One that we haven't mentioned is something that used to be a huge problem as well, and that's conjunctivitis in newborns born to women who have gonorrhea. That used to be a major cause of blindness in newborns. So that's another consequence of gonococcal infection that we would like to be able to avoid seeing increase in incidence in the situation of untreatable gonorrhea.
Fantastic. So, Bill, you were starting talking about the resistance. Mm-hmm. So we've had several drugs throughout history, as gonococcal is as probably as old as human beings themselves. But lately, the main concern have been with the rise of resistance to the fluoroquinolones antibiotics, as was part of the guidelines initially, older guidelines, and of course now the beta-lactams. So, can you tell us a little bit about the mechanisms by which the gonococcus become resistant to these drugs?
Sure. Let's start off with the beta-lactams. So, there's essentially two mechanisms of resistance. One is that develop very slowly over time, multiple chromosomal mutations that impact drug influx, and those are mutations that affect the major outer membrane porin protein, and then mutations that occur in penicillin binding protein 2, which would be equivalent to penicillin binding protein 3 in E. coli, and a single mutation in penicillin binding protein 3. So the other important component of that is there's a multi-drug efflux pump called MTRCDE that can recognize beta-lactams, and mutations develop that impact the transcriptional activity of the operon that encodes the pump. So that occurred over a long period of time. When I was a graduate student in the mid-70s, I got fascinated about the gonococcus when I was working on staphylococcus because there was a couple of papers that came out about the beta-lactamase plasmid that found its way into the gonococcus. So that gives a very high-level single-step resistance. But what shut down the use of penicillin in the United States in the mid-1980s was that first system that I told you about, the slow buildup of mutations. And then what happened, the gonococcus can receive DNA from commensal Neisseria. So there were a couple of events that occurred that I think are really important, especially with the third generation cephalosporins. They received DNA from one of the commensals, and that caused a mosaic structure of the penicillin binding protein 2 encoding gene. There's about 64 amino acid changes. Also, too, they received DNA that was inserted into the locus that encodes the transcriptional regulator of that drug efflux pump, as well as in the pump operon itself. So you had this situation over time where we can appreciate that mechanisms of resistance can be spontaneous mutation or gene acquisition or both. So that's, in a nutshell, how beta-lactam resistance occurred. And there are probably other mechanisms that impact MICs. So, and as we learn more about the genome, and maybe Yonatan can talk about this, that we're going to uncover other mechanisms that either decrease intake, increase efflux, or modulate some other physiologic function. And then with the fluoroquinolones, I think we have to go back to the early 1990s with some very pioneering work done by Bob Bellin when he was at the Rocky Mountain Labs. And this is when Cipro was being used, and he wanted to know how could the gonococcus develop resistance to Cipro. And basically what he found is that there were two mutations in the gyre gene, called DNA gyrase A, at positions 91 and 95. And then there was a second step to give higher level resistance in the topoisomerase subunit PAR-C. So you bring those together, and you can get high-level resistance. And that, from just a historical trend, that happened quicker than the beta-lactam resistance, because there were essentially 3 mutations that could occur. And again, there's probably, and Yonatan can speak to this because his lab just published a real elegant story that about JAR A and PAR C. There's probably other mutations that can occur that can provide a clinical level of resistance.
So, Yonatan, why don't you tell us about the story? You know, we are all dying to hear.
Well, thank you. And thank you, Bill, for kind words. So maybe what I'll do is mention a couple of things. First, on cephalosporin resistance, a quick story. And this is work by Samantha Pallas, who's a postdoc, now staff scientist with me here in the lab, where we were interested in looking at a few isolates that had ceftriaxone resistance, but didn't have the mutations in PEN-A, which is the gene that codes for penicillin binding protein 2 that you heard Bill talk about. We didn't find basically a reason why these isolates should have the increased MICs to ceftriaxone that they did. And through a combination of both computational and experimental work, Sam was able to identify that they had mutations in RNA polymerase subunits, RPOB and RPOD, each of which seemed to independently confer higher level resistance to ceftriaxone. And we still don't understand the mechanism there, but she was able to show very cleanly that these mutations can confer resistance.
It is interesting that RPOB and C, for example, have been associated with daptomycin resistance. So definitely this mutation changed the envelope somehow in these organisms, which is really fascinating.
I totally agree. And it's a story that, interestingly, similar kinds of mutations have been reported even in Enterococcus. RNA polymerase that increase resistance to some of these cell wall acting agents. And unclear exactly how they work. For the, I'll mention actually a couple of stories related to quinolone resistance, because we've an MD PhD student who just recently defended in the lab, Dan Rubin, published a couple of papers, one in Nature Microbiology and the other in Lancet Microbe, that deal with quinolone resistance. One of the first things we were interested in was why some lineages seem to acquire and maintain resistance better than others. We think of resistance mutations as incurring a fitness cost, and so some strains may be better able to accommodate that fitness cost than others. We think about sometimes compensatory mutations as happening in sequence. First you acquire resistance, then you acquire the compensatory mutations. But it may well be that some strains actually have enabling or stepping stone mutations that facilitate the acquisition of resistance. And one of the things that Dan did was to show that this was actually the case, and there was a basic metabolic gene called the beta-carbonic anhydrase, or we termed it CAN-B for Neisseria gonorrhoeae, and that one amino acid mutation in this gene really determines whether the Neisseria gonorrhoeae requires supplemental CO2 in order to grow. And this also facilitates, you have this mutation, it facilitates the acquisition of resistance to fluoroquinolones. The paper I think that Bill was referring to, the more recent one in Lancet Microbe, was one related to, I think, another topic that you wanted to talk about on this podcast, which is diagnostics. One of the things we didn't talk about when going over the clinical manifestations of Neisseria gonorrhoeae is how we approach it clinically. So if it's okay, I'll take a moment just to mention how we do that. Most cases of gonorrhea are diagnosed by nucleic acid amplification tests. These tests are sensitive and specific, but they just tell us that Neisseria gonorrhoeae is there. They don't tell us anything about the antimicrobial susceptibility profile. For that, we have to do culture and grow them, and that takes a long time, and very few labs continue to do that. So for many isolates, we just don't have the information. What people have been trying to do is develop diagnostics to go from genotype to phenotype. since we're already looking by nucleic acid amplification tests, can we find signatures of antibiotic resistance or susceptibility? And the most success in this regard has been in finding a diagnostic for quinolone resistance. As Bill described, mutations in gyrase A, are really where we see a clear correlation. Like codon 91, if you have serine, the isolate will be susceptible. If it has a phenylalanine, it will be resistant. So just from that single codon, you can, with very high confidence, predict susceptibility and resistance. And there's a diagnostic now that's been approved in Europe, in the UK, in Australia, New Zealand, and I think is under expedited consideration by the FDA here in the US, to look at exactly this locus. And I think a bunch of companies are trying to make diagnostics looking at it too. If we can use Cipro, it would be great. It's a good drug, and only about 30-35% of isolates in the US are Cipro resistant. So it would be adding to our options for treatment. In this paper, what we were interested in asking about was the selective pressure that we would apply through this diagnostic. So whenever you use a drug, you're applying a selective pressure. In this case, you're using a drug targeting isolates that have this gyrase 91S. So we were curious, could it develop a diagnostic escape where it would find an alternative pathway to resistance? Because that's basically what we're selecting for using this diagnostic and guiding treatment through it. And we did this in the lab. We took a bunch of isolates from around the phylogeny, and we first asked if we converted a 91F or phenylalanine to a 91S, what would we find? Would we find isolates that still had non-susceptible MICs? And the answer was yes. There were some isolates that even when we convert the 91 to a 91F to a 91S, the MICs dropped a lot, but they were still in the intermediate category, non-susceptible category, where we would expect some treatment failures. We then asked, okay, they'd be under a selective pressure, and using experimental evolution in the lab, would we see resistance emerge through some pathway? And we did. While maintaining the 91S, which we normally associate with susceptibility, these isolates developed mutations in GyrB, another component of the replosome, that conferred resistance. And those GyrB mutations, one of them had already been described in Salmonella as part of, as conferring quinolone resistance. So it seemed like a quite reasonable thing to think that it might also happen in gonococcus. And another one, which we totally didn't expect, is identified as something conferring resistance to zoliflodacin, which I think will come up in conversation a little bit, because it's a drug we haven't even used yet. It's a new drug that's in phase 3 trials for treatment of gonorrhea. So our point in this paper was that diagnostics can be great, but we should also expect that they will apply selective pressure, and sometimes those can yield unexpected or unanticipated consequences.
Great stories. Thank you for sharing those. So before we move sort of more therapeutic, Bill and Yonatan, you can complement that. So what do we know about the population structure globally of the gonococcus? I mean, we know that there are certain lineages in different other bacteria that are more prone to acquire mutation and cause disease. What do we know about this in gonococcus?
I'm going to defer to Yonatan because he's been actively involved in the genomics and bioinformatics and has a fantastic story to tell.
Well, as have you, Bill. So it's all stuff you can talk about as well. But well, I think one thing here that's also important to note is Neisseria gonorrhoeae is highly recombinogenic. So it actually not only exchanges DNA with other Neisseria, leading to the mosaicism that Bill referred to at the Pen-A locus and at the MTR-CDE, MTR operon, but all over. So there's extensive recombination. So looking across the species, when you're trying to go back in history and reconstruct history using shared mutations, it becomes a little bit of a mess pretty quickly because of the extent of recombination. That said, we definitely see lineages spread pretty quickly and spread all around the world. This is one of the remarkable features of gonorrhea, going back to our inferences about its origins. It looks like there's a beautiful paper by Leo Sanchez-Busso, who's then at the Wellcome Sanger Institute, where she used a global collection of Neisseria gonorrhoeae to try to ask questions about its origin. And she inferred that it originated the time to the most recent common ancestor. When was that? Well, she estimated that it was in the 16th century. Right? So far more recent than people might have expected.
I didn't expect that. Yeah.
Yeah, I had to actually change my introductory slide because I was quoting the book of Leviticus
Yeah.
to,
Like syphilis, right? Like,
for the Israelite women to avoid men with discharges. And then I read that paper and I said, okay.
Yeah, so I think it's a totally interesting point. And gonorrhea was named by Galen, right? 2,000 years ago. So I think there was a gonorrhea-like syndrome that existed way back then. One of the things that we learned when working with the CDC and sequencing all of these samples derived from men with this syndrome was that the vast majority were caused by Neisseria gonorrhoeae, but we also found isolates that were from Neisseria meningitidis and Neisseria lactamica. So here were these other close relatives of Neisseria gonorrhoeae causing what to all the world looks like gonorrhea, but caused by another related species. So what do all of these have in common? They all hang out in the oropharynx. Right? That's where you find all the commensal Neisseria, and that's where Neisseria gonorrhoeae originated. And the idea is that probably, this activity, opportunity to enter into the urogenital region happens with some frequency, and that many of these Neisseria may have caused a gonorrhea-like syndrome dating back to Leviticus, dating back to Galen. But what happened around the 1500s that would have allowed a new species to really find its niche and expand? Well, around that time, the population in Europe was growing. Mercantilism, right? This was the time of the commercial revolution. Mercantilism was expanding, and it was a time of increased maritime travel. Right? This was really when sailors started exploring the world. Around the same time, and you mentioned syphilis, Cesar, in just a brief allusion to it. Its first appearance was 1494 at the Siege of Naples. Right? So around the same time, in around the same place, we think we see the emergence of two STIs. Totally interesting. And I guess, hypothesis, would be interesting to try to dig into this, is that it had something to do with the ability of the sexual contact networks to support transmission of STIs, right? The density of the networks, the extent of connection among individuals. So when we get to the reproductive number, a lot of it is about contact rate. And so perhaps the contact rate was now high enough to be able to sustain population-wide spread of these. But one of the most interesting things in Leo's paper was that the origins, which seem to be in Europe, there was a branch that quickly went off very soon afterwards to Asia. And then there was circulation in Asia. And that makes sense. Like going back to this maritime idea, the Dutch East India Company, for example. There's certainly a lot of movement back and forth. And of course, sailors, right? I mean, that's a classic kind of trope for STIs. So this global spread is something that we have seen from its very beginning, and we see it now. The isolates that I think we'll probably talk about later that carry ceftriaxone resistance that have appeared in the UK, in the US, elsewhere in the world seem to be originating in Asia. But these are lineages that then spread globally. So they appear in one place, they can find a fitness niche, and then they're able to get into these networks and spread. So we do see dominant lineages, but they turn over as perhaps our drug use changes. And there were these two lineages going back a long way, one which ended up being more in different sexual networks, so men who have sex with men, and then also in heterosexual populations. And More recent studies of that, including a beautiful one by Deb Williamson in Victoria in Australia, showed that there's now extensive bridging across sexual networks that you can see from these phylogenies. So we're really seeing now a lot of mixing among the sexual networks and internationally.
Yeah, and
what a
Cesar,
fascinating story.
yeah,
Yes, Bill.
if I could follow up on that, more recently, I think something fascinating happened in 2012 when the treatment guidelines were changed from cefixime to ceftriaxone and azithromycin. To me, azithromycin, for a variety of infections, has been one of the most abused antibiotics. And what we saw happening is that after 2012, the percentage of strains with decreased susceptibility to azithromycin just went off the charts. In just a few years, the CDC removes an antibiotic from the treatment regimen when the prevalence is 5% or greater. And so what happened was, I think, that all of that pressure by azithromycin triggered recombination events with the commensals. Okay. And a big part of that resistance, which was, 2 micrograms per mil or greater, was due to recombination in the MTR locus, where you had this mosaic structure being formed, where you had increased expression of the pump plus mutations in the major transporter. So as soon as you put a new antibiotic pressure on the bug, there's going to be new events that occur, probably recombination with commensals.
So this is a perfect segue to then spend the last time of the show talking on treatment, right? So, Yonatan, how do you treat 2023, March of 2023? How do you treat a patient, let's say, that is native to the United States versus a patient that maybe comes from Asia or has traveled in Asia and has some sexual contacts there? So how do you approach those patients?
Well, in the US, the CDC guidelines are to treat with 500 milligrams of ceftriaxone intramuscularly times 1. Right? It's a single dose of intramuscular ceftriaxone. That's in the US. The treatment guidelines are different in other places. In the UK, it's 1 gram, which it's about as much as you can give, right? I mean, that's going to be difficult for patients to sit down for some time after getting a gram of ceftriaxone. Right? And then in Australia, they still have, it's my last checking, dual treatment still with azithromycin. So it depends really on where you are for treatment. But in the US, 500 milligrams. And we haven't seen treatment failures yet. So just recently, in late January, the Massachusetts Department of Public Health reported the first cases of isolates with either resistance or reduced susceptibility to all of the antibiotics that we use for Neisseria gonorrhoeae. That includes isolates that have this a particular allele at pen A. It's called pen A60, but it's one that we associate with reduced susceptibility to ceftriaxone, the highest high levels of ceftriaxone resistance. But this patient was actually cured by the treatment that he received with 500 milligrams of ceftriaxone. Now, where we have seen treatment failures globally with these kinds of isolates, it has been in patients who have pharyngeal infections. For reasons I have to say I don't understand, and maybe Bill does, pharyngeal infections are much harder to treat than infections at the other mucosal sites, or even bloodstream infections. Pharyngeal infections are just difficult to clear with these single dose therapies. So, the rates of success with ceftriaxone are a little bit lower, still very high, but a little bit lower. Rates of success with azithromycin, any of the other drugs, they just are decreased for pharyngeal infection. In the UK, where they've seen these treatment failures with ceftriaxone, the ones that have reported have all been pharyngeal infections. So something about that is, interesting biology to be explored. Perhaps PKPD, perhaps something about the biology of the organism, perhaps something about the microbiome. I'm not sure. The last thing that I'll mention here is, that you mentioned about how we approach these kinds of cases for people who have recently traveled or so on. But the Mass DPH, Massachusetts Department of Public Health, recommended after these cases that really clinicians should start getting cultures from their patients. And I think it's a really interesting and important step that will help us get a better understanding of what's circulating, and that perhaps some of these strains may well be out there. This person who was identified as having this strain did not report any recent travel, right? So there's concern that some of these strains may actually be under the radar circulating already, right? So we need better surveillance to really be able to detect it. We don't know kind of how deep the iceberg goes.
Mm-hmm.
So, you get this report from Asia and other countries, and this is bound to be seen here in the United States at some point. And that's the reason the impetus potentially to develop new drugs. And actually, fortunately, we have some new drugs there in the pipeline that are probably close to the finish line, hopefully. So let's talk about those. So what new drugs are available for gonococcus? Bill, you want to start?
Well, what's coming down the pipeline? I mean, you've mentioned one of them, and another one Yonatan mentioned. And we can talk about zoliflodacin. There's a phase 3 clinical trial. It shows great promise in treating urogenital tract infections. Like other antibiotics, it had an issue with oral treatment. We need more data on efficacy in women from that trial. The thing that's fascinating to me is there's a few papers recently published that has looked at the potential for Neisseria gonorrhea to develop resistance. And just screening thousands of isolates, resistance is very, very low. So that's good news. But there's a fascinating, and I reread this paper last night, there's a fascinating isolate from, I believe it was Japan in 2000. So zoliflodacin hasn't even been entered, massively throughout the world. And this strain already has a mutation that we know can confer resistance.
So what's the mechanism of action?
I'm sorry. So the mechanism of action is at DNase gyrase B subunit. So I think that there's going to be the potential for that GyrB mutation to further be selected globally. And, as I mentioned, every antibiotic that's been brought into clinical practice and used massively, sooner or later, you're going to see resistance. So even though it's not seen now and the initial trials seem promising, it could be a replacement drug or it could be an additive drug that can be kept in the arsenal. And then, there's others that could be further coming down the pipeline.
Yonatan?
Yeah, I think that that's right. I think that the ability for Neisseria gonorrhoeae to explore its evolutionary landscape and find the right combination of alleles that allow it to acquire resistance while maintaining fitness suggests that if we just use zoliflodacin alone, we should expect to see eventually these resistant strains will emerge and spread. And you mentioned the resistance in Asia. In fact, ceftriaxone resistance, as reported by a few countries in Asia, is already fairly high. And you're right, we'll start to see that spread around the world. And I think in a report from the UK just this past fall, the 10 isolates that they describe of ceftriaxone resistance, the majority of them were from individuals who had traveled to Asia. So I think you're right. That's where we're seeing a lot of the selection happen and then spread. And zoliflodacin, I think, does show promise, and it has that same issue as Bill mentioned, that it doesn't do, it seems, at least in the reports that I've seen from the phase 2 trial, isn't great at clearing pharyngeal infection. Not as good, at least, as ceftriaxone. Gepotidacin is the other drug that's in phase 3 trials. In its phase 2 trial, and it also targets the replosome. So like zoliflodacin and fluoroquinolones, although they all work slightly differently. Zoliflodacin and gepotidacin are in each first in class, 2 different classes of antibiotics. But in the phase 2 trial for gepotidacin, out of 69 individuals, 3 developed resistance on treatment. 2 of them seemed to have kind of stepping stone mutation that facilitated the acquisition of resistance. But this certainly alarmed a number of people, and GSK, that makes the drug, has decided that, in fact, instead of a single dose treatment, they're going to use a 2-dose regimen in their phase 3 trial, where it's 2 doses, 1 dose taken 10 to 12 hours or so after the first dose, as a way to try to limit the emergence of resistance. So really trying to make a PK/PD argument for resistance suppression here, and changing the paradigm in some sense by making it into, instead of a single dose therapy for gonorrhea, into a 2-dose therapy. So gepotidacin, like zoliflodacin, also had treatment failures in pharyngeal infection. So I think that's going to continue to be, something we scratch our heads over in terms of how best to deal with it.
So beautiful. I learned so much today. So let's conclude. So what other things in terms of treatment, are common? Remember, there are phages that seems to be interesting. Some virulence modulator agents. So is there anything exciting going on in the gonococcal world in that realm?
A couple of things I think that are exciting is we've had a resurgence in interest in developing a vaccine. Back in the 80s, there was a big emphasis of developing a vaccine against the pilus because it's needed for attachment. If you block attachment, maybe you could block infection. Well, at that time, in the early 1980s, we didn't know about antigenic variation. We knew that there were different colony types that were associated with pili, and the pilus vaccine trials, you can go back and read it, they failed. So interest in the vaccine dropped because antibiotics were still very effective and against the gonococcus, and we sort of put it off to the side. And then, a few years ago, when it became evident that we could have an antibiotic resistance issue, the interest in developing vaccines had a reemergence, and there's several groups working on that. The other, interesting areas are maybe targeting certain steps in resistance that are important. Like maybe if you could inhibit efflux, you could keep the concentration of the drug higher in the cytosol or in the periplasmic space. And there's a resurgence in doing that in gram-negative bacteria. So we could enter that. We might be able to target down the road, certain essential genes. So I think that there's a lot that can still be done from a basic science point of view. And I used to think that we would never have a vaccine against gonorrhea, but my thoughts have changed now because we know more about human immunology and we know more about adjuvants. So that's one area that I would say we should keep on our radar screen.
Yonatan, the last word, futuristic view, your crystal ball.
Oh, I really hope we can move forward with a vaccine. I think that would be great. You mentioned phage therapy. I guess we've tried to isolate phage for Neisseria gonorrhoeae. We haven't had any success. So, and interestingly, it doesn't have a CRISPR-Cas system. So it has some way of dealing with phage, but also is a little bit of a mysterious area that's going to be interesting to delve into more. We didn't talk about doxypep or post-exposure prophylaxis using doxycycline, but that's an area therapeutically that has, well, really as a preventative, has garnered a lot of interest since reports from a few trials using doxycycline for post-exposure prophylaxis have started to report results. But that, I think, is also going to be a very interesting shift in how we approach STIs broadly. But the impact on gonorrhea, I think, is going to be one of reshaping the gonococcal population, where we're applying a steady pressure from doxycycline. So I think there's going to be there's much exciting work to be done in vaccines, in understanding the basic biology, and also continuing to understand how the pressures that we apply as healthcare providers, individuals, how those shape the gonococcal population both here and globally.
Phenomenal. Thank you very much for this wonderful conversation. This has been really amazing. I really appreciate that. So this is Cesar Arias, Editor-in-Chief of AAC, signing off.